All right, so we are studying cell organelles, and let's start with lysosomes. Lysosomes are lysosomes are sites of intracellular digestion and turnover of cellular components. So this this just the quick function of lysosome. These are the sites of intracellular digestion and it's responsible for turnover of cellular components. Lysosomes, as the word explains, lysis, solution, plus soma means body. So lysis is solution and plus soma meaning body. Are membrane limited vesicles that contain about 40 different hydrolytic enzymes and are particularly abundant in cells with great phagocytic activity. So what does lysosome? Lysosomes basically is a site which contains about 40 different hydrolytic enzymes. Hydrolytic means water loving and are particularly abundant in the cell with great phagocytic activity. Abundant in cells with great phagocytic activity. We have read the great phagocytic activities are macrophages and neutrophils when we studied the phagocytosis. Although the nature and activity of lysosomal enzymes vary depending on the cell type, the most common are acid hydrolysis such as proteases, nucleases, phosphates, phospholipases, sulfates, and beta glucuronidase. So we know whenever we have a suffix aces means it is a digesting hormone or a protein or which cleaves something. For example, protease cleaves protein, nucleases cleave nucleus, phosphatase cleaves phosphate and separates it from its group. Phospholipases is the one that ligates phospholipids. So as can be seen from the list, lysosomal enzymes are capable of breaking down most macromolecules. As I said, break down, lysis. All right, lysosomes, which are usually spherical, range in diameters from 0.05 to zero point 0.5 micrometers and present a uniformly glandular, electron dense appearance in the stem. I don't think we need to remember the appearance of the lysosomes. We will see them in the electronogram that is in, let's check, figure 2.16. Yes, so here in figure 2.16 we have a cell. G represents Golgi and L is the lysosome. L here is lysosome. These are the sites where we have abundant of about 40 different types of hydrolytic enzymes. All right. So in macrophages and neutrophils, lysosomes are slightly larger and visible with the light microscope, especially after histochemical staining. Cyto Cytosolic components, that is cytoplasmic components, are protected from these enzymes by the membrane surrounding lysosomes and becomes the enzymes have and because the enzymes have optimal activity at an acidic pH of about 5. Any leaked lysosomal enzymes are particularly inactive at the pH of cytosol, that is 7.2, and harmless to the cell. So we need to remember that lysosome, these 40 hydrolytic enzymes are covered in a membrane. Membrane is important to have an acidic atmosphere inside for these enzymes to be active. pH of the cytoplasm is 7.2 and these enzymes are just useless in the cytoplasmic environment. Lysosomal hydrolysis, we can just remember the optimal activity pH data just in case if it's a question some university asks them. So lysosomal hydrolyses are synthesized and segregated in the rough endoplasmic reticulum and then transferred to Golgi apparatus, where the enzymes are further modified and packaged in vacuoles that form lysosomes. So basically, we know that the hydrolyses basically are proteins, right? So all these nucleases and proteases and phosphates, these are all proteins. So how the polypeptide chain is produced on the ER and then goes through Golgi, same goes with these enzymes. The maker's mannose 6 phosphate M6P is added to the phosphotransferase in the cis Golgi only in the N-linked oligosaccharide for the hydrolysis designed by the lysosomes. So the marker here is MP6 that is added to phosphotransferase in the cis phase Golgi. This is a part of packaging. N-link means nucleotide link oligosaccharide of the hydrolysis destined for lysosomes. So membrane receptors for MP6 containing protein in the trans-Golgi membrane then bind this protein and divert them from the secretory pathway to the segregation into lysosome. So this packaging is important for the identification. When we have a marker M6P, the trans phase of the Golgi knows that this has to be packaged in a form of a secretory in a form of vesicle, and through a secretory pathway these proteins are segregated into the lysosomes, which protein which have a marker M6P. All right, material taken from the outside the cell by endocytosis is digested when the membrane of the phagosomes or pinocytotic vesicles fuse with a lysosome. All right, so this is the important function because we need to digest the the phagosome and the pinocytotic vesicles, and this happens with when they fuse it with the lysosome. The first thing that happens is they go and fuse with lysosome. This mixes the endocytosed material with the lysosome material with the lysosomal enzyme and activates proton pumps in the lysosomal membrane that acidifies the content, allowing digestion. So like the digestion happens in our stomach, we have acid, and the process, we need acidic environment to start the process of digestion. This acidic environment is given by lysosome. So any phagocytotic substance or pinocytotic substance comes into the body, first goes to the lysosome, the membrane is ruptured and it's ready for digestion in the acidic environment provided by lysosome. The composite active organelle is now termed as secondary or heterolysosome. So we need to remember what is heterolysosome, the one that is involved in this digestion process, the one that has accepted this phagocytosed molecule. Heterolysosome are generally somewhat larger and have a more heterogeneous appearance in the temp because of wide variety of materials they may be digesting. So of course we have an extra material added to the lysosome, it will appear larger. During this digestion of macromolecules, released nutrients diffuse into the cytosol through the lysosomal membrane. During this digestion of macromolecules, released nutrients diffuse into the cytosol through the lysosomal membrane. Yeah, of course. How do they diffuse? Through the membrane. Indigestible material is retained within a smaller vacuolar remnant called residual body. Yeah, so whatever that is indigestible by the lysosome, it is separated in the form of residual body. We can just highlight what are residual bodies in terms of lysosome. In some long-lived cells, residual bodies can accumulate over time as granules of lipofuscin. Yeah, so lipofuscin is something important. We have read about lipofuscin before. It is going to help us for cell apoptosis at some point. Uh, let's keep it in this way and we'll read about this much in detail. In some long-lived cells, residual bodies can accumulate over time as granules of lipofuscin. All right. So besides degrading exogenous macromolecules, lysosomes also function in a process called autophagy, by which the excess of organelles or large aggregates of non-functional macromolecules in cytoplasm are degraded. So we know what autophagy is cell death, programmed cell death. During autophagy, a lipid bilayer membrane forms around and isolates the cytoplasmic portion to be removed, producing an autophagosome. So, autophagosome is basically a portion that is going to be removed by the process of autophagy. A lipid bilayer membrane. All right. Which then fuses with the lysosome for digestion of the enclosed material. Of course, autophagy is enhanced in times of starvation or nutrient stress. Amino acid, nucleotides, and other digestive products are released from the autophagosomes for reuse as a source of energy and in newly synthesized reaction. So that's it. Nothing much here, and let's go to proteosomes. Proteosomes are very small, abundant protein complexes that are not associated with membrane, each approximately the size of the small ribosomal subunit. So basically, it's very small, like a ribosomal subunit, and these are complex that are not associated with membrane. So proteosomes are non-membranous. They function to degrade denatured or otherwise non-functional polypeptide. We need to know the function of proteosomes because they are responsible for degradation of non-functional polypeptide, means non-functional proteins. Proteosomes also remove proteins no longer needed by the cell and provide an important mechanism for restricting the activity. So we need to differentiate very specifically, very importantly, that both lysosome and proteosomes are responsible for degradation, but mostly lysosome is responsible for digesting the material that is being accepted through exocytosis, and only autophagy is a phenomenon where it degrades all the organelles and the cytoplasmic content of the cell, causing eventually cell death. But in case of proteosome, they will never cause cell death. Only the non-functional proteins, the non-functional polypeptides are degraded. Proteosomes also remove protein no longer needed by the cell and provide an important mechanism for restricting the activity of specific protein to a certain window of time, whereas lysosomes digest organelles or membranes by autophagy. Proteosomes deal primarily with free proteins as individual molecules. So this is the difference I explained. Whereas lysosome digests organelles or membranes by autophagy, it's going to digest membranes by autophagy. Proteosomes deal primarily with the free proteins. Let's highlight this difference so that we can revise this next time. As shown in the figure 2.9, we'll check figure 2.9. The proteosomes in a cylindrical structure made of four stacked rings, each composed of seven proteins, including proteases. All right. At each end of the cylinder is a regulatory particle that contains ATPase and recognizes protein with attached molecule of ubiquitin, an abundant cytosolic 76 amino acid protein found in all cells. So, what is ubiquitin? Is 76 amino acid protein found in all cells. At each end of the cylindrical, there is a regulatory particle that contains ATPase. All right. Misfolded or denatured protein or short-lived protein with oxidized amino acids are recognized by the chaperones. The recognition of this, we also saw the role of chaperones in the when we read about the endoplasmic reticulum. It is responsible to figure out and find out the non-functional protein, the proteins that are unfolded and eventually become unstable. Chaperones is responsible to segregate them, to find them, and targeted for destruction by other enzyme complexes that conjugate ubiquitin to lysine residues of the protein, followed by formation of polyubiquitin chain. So after the recognition by, let's also highlight this. So we have recognition by chaperones, and then these polypeptides are targeted for the destruction by other enzyme complexes. What are these enzyme complexes? Which contains ubiquitin to lysine, eventually forming a polyubiquitin chain. Ubiquitinated proteins are recognized by the regulatory particles of proteosomes unfolded by the ATPase using energy from ATP and then translocated into the core of the cylindrical structure and degraded by endopeptidase. So endopeptidase is an enzyme that eventually degrades the protein. The ubiquitin molecule are released for reuse and then peptide. So ubiquitin is required to form this complex where a protein can be brought by the identified by the chaperone, brought into a complex by ubiquitin, and endopeptidase is the enzyme that eventually degrades this protein. So the ubiquitin molecule are released for reuse. After this process is done, the ubiquitin molecules go back reuse, and the peptide produced may be broken down further to amino acids, or they may have other specialized destinations, such as antigen presenting complexes of the cell activating an immune response. So this specialized destination is important just in case we know that the broken polypeptides can just be in the cytoplasm in the form of amino acids, or they can have some immune response. They can be responsible for activating some kind of immune response by the cell. So this was proteosomes. All right, so let's get into mitochondria. Mitochondria, as the word explains, mito means thread and chondria means granule, are membrane enclosed organelles with arrays of enzymes specialized for aerobic respiration and production of adenosine triphosphate. With high energy phosphate, we need to remember this membrane enclosed organelle, because this is the question that is frequently asked and tried to this type of questions try to confuse students that, oh, which organelle was membrane bound, which was not. Mitochondria is membranous organelle, and it, with so there are membrane enclosed organelles with arrays of enzymes specialized for aerobic respiration. Guys, remember this is an aerobic respiration and production of adenosine triphosphate, that is ATP, that is energy, with high energy phosphate bonds, which supplies energy for most cellular activity. Glycolysis converts glucose anaerobically. Glycolysis is a process anaerobic, so basically happens in cytoplasm. Anaerobic to pyruvate in the cytoplasm, releasing some energy. It does release some energy, but not enough. It's not a complete energy product. The rest of the energy is captured when the pyruvate is imported into the mitochondria and oxidized to carbon dioxide and water. Mitochondrial enzyme yield 15 times more ATP than is produced by glycolysis alone. We need to remember this data 15 times. This, whenever we're reading about glycolysis and aerobic respiration, they are going to ask for this data like 5 times energy than glycolysis, and the number of ATP produced in the process from glycolysis and aerobic respiration, and so on. We'll read about these numbers here. Some of the energy released in the mitochondria is not stored in the ATP but is dissipated as heat that maintains body temperature. So we lose some in this whole reaction, we lose some energy in the form of body temperature. Mitochondria are usually elongated structures with diameters of 0.5 to 1 micrometer and lengths of up to 10 times greater. They are highly plastic, rapidly changing shape, fusing with one another and dividing, and are moved through the cytoplasm along microtubules. So microtubule is a part of is a type of cytoskeleton, and like other organelles, this microtubule helps the mitochondria to stay in shape and to move around in the cell. The number of mitochondria is released is related to the cell's energy need. So whenever cells where we require more energy, we have more mitochondria. Cells with high energy metabolism like cardiac muscle cells or cells of kidney tubules, have abundant mitochondria, whereas cells with low energy metabolism have low mitochondria. This example, this just example are important, that cardiac muscle cell or the kidney cells are high mitochondria. Similarly, mitochondria of differentiated cells are considered in cytoplasm are concentrated in cytoplasmic regions where energy utilization is more intense. Of course. All right. Mitochondria are often sufficiently large to be visible with the light microscope as numerous discrete organelles. Check figure 2.19. Under the thumb, each mitochondrion is seen to have two separated and very different membranes that together create two compartments, the innermost matrix and a narrow intermembranous space. So this is very, very important, that when seen under the thumb, it has two separated membranes, very different membranes. One, two compartments. The innermost is the matrix and a narrow intermembrane space. Both mitochondrial, I will explain this in the figure 2.19. So, oh, figure 2.19 does not, oh, okay, we have it here. So this, remember guys, this is the intermembrane space and this here is the matrix. I hope you could understand and see it. All right. Both mitochondrial membranes contain a higher density of protein molecules than other membranes in the cell and have reduced fluidity. So, yeah, so both membranes have protein molecules which eventually reduce fluidity. The outer membrane is sieve-like, containing many transmembrane protein called porins that form channels through which small molecules such as pyruvate and other metabolite readily passes from the cytoplasm to the intermembranous space. We need to remember about these porins. The inner membrane has many long folds called cisternae. Cristae. You know, I misspell this word, sometimes as a when I was reading this as a kid, I always used to, misspell it, and the misspelling has stuck in my brain. But it's called cristae. Cristae. The inner membrane has many long folds called cristae, which project into the matrix and greatly increase the membrane's surface area. So cristae, as we know, we had this in the ER as well. They are structures basically responsible and is important where we need greater surface area. The number of cristae in mitochondria allow corresponds to the energy of the cristae in mitochondria. Sorry, the number of cristae in mitochondria allows also corresponds to the energy needs to the cell. The lipid bilayer of the inner membrane contains unusual phospholipids that and is highly impermeable to ions. We need to know the impermeability of the ions, the lipid, because the lipid bilayer of the membrane is impermeable to the ions because it contains unusual phospholipids. Integral proteins include various transport proteins that make the inner membrane selectively permeable to the small molecules required by enzymes in the matrix. Of course, so we need some molecules in the matrix for the aerobic respiration process, and we need a selective protein there. Mitochondrial matrix enzymes include those that oxidize pyruvate and fatty acids to form acetyl coenzyme A, and those of the citric acid that oxidizes acetyl coenzyme A release CO2 as waste and small energy rich molecules that provide electrons for transporting along the electron transport chain. Super, super important. What does the mitochondrial matrix enzyme consist of? They consist of enzymes that are responsible to convert fatty pyruvate and fatty acids to form acetyl coenzyme A. This is what is going to be later converted, oxidized into forming CO2 and energy. As waste, and small CO2 is going to be the waste product here, and a small energy rich molecule that provide electron for the electron transport chain is produced. Enzymes and other components of this chain are embedded in the inner membrane and allow oxidative phosphorylation, which produces most of the ATP in the animal cell. So oxidative phosphorylation means, the word suggests removal of the phosphate group, and oxidative means in the presence of oxygen, ATP are produced. Let's see how and what exactly happens. Formation of ATP by oxidative phosphorylation enzymes occur by a chemoosmotic process. It's a chemoosmotic process, means chemically regulated. Oh god, what's wrong? All right. Membrane proteins guide the small electron carrier molecule through closed packed enzyme complex so that the electron moves sequentially along the cell. So this membrane protein guide this electron, and the electron moves through the electron transport chain that is on throughout the membrane. Electron transfer is coupled with oriented proton uptake and release, with proton accumulating in the intermembrane space and producing an electrochemical gradient across the inner membrane. So as the electron moves, a H plus is pumped into the intermembrane space. We know what is the intermembrane space and the matrix. Matrix is different, intermembrane space is different. So super important here, that this proton pump, the presence of proton in the inner membrane space eventually creates an electrochemical gradient, basically the concentration gradient. Membrane associated proteins of the ATP synthesis system form large 10 nanometers multi-subunit globular complexes on stalk-like structure that projects from the matrix side of the inner membrane. Check figure 2.20. So here is the figure 2.20. As I said, this is the inner membrane space where we have the hydrogens present. These are the electron transport complexes, blue in color. And when an electron comes and moves throughout this chain, this hydrogen from the matrix is pumped into the electron transport into the intermembrane space. But remember guys, there are four complexes attached. First, third and fourth ones are the one that pumps the protein, and second complex does not pump the protein. So it's a lot of biochemistry here, but we need to remember that the second complex does not pump hydrogen. We will probably read it here. Membrane associated proteins of the ATP synthetase system. All right, we read this. This enzyme complex contains a channel through which protons flow down the electrochemical gradient, crossing the membrane back into the matrix. So simple. When we have a high concentration of hydrogens in the intermembrane space and low in the matrix, there is a gradient developed, and they move through this intermembrane from higher concentration to the lower concentration. So from the intermembrane space to the matrix, eventually producing the ATP. So let's read how does this happen. These enzyme complexes contain the channel through which the proton flows down the electrochemical gradient, crossing the membrane back into the matrix, as I said. Passage of the protons through this channel causes rotation of the specific polypeptide in the globular ATP synthetase complex, converting the energy of proton flow into the mechanical energy of the proton movement. As I said, this movement of the proton is a chemical energy that, and it flips the protein on the movement, and this protein movement even is basically the mechanical energy. Mechanical energy stored in the new phosphate bond of ATP by other subunit polypeptide binding adenosine diphosphate. So this adenosine diphosphate in this reaction, when the proton pumps in the matrix, what exactly happens on those membrane proteins is that ADP and a phosphate group is added to this ADP, eventually forming ATP. A steady torrent of the proton along the gradient allows each of these remarkable synthetase complex to produce more than 100 molecules of ATP per second. We got to remember is this number: 100 molecules of ATP per second are produced. Another role for mitochondria occurs at times of cell stress when the protein cytochrome C is released from the inner membrane, the electron transport chain. Okay, so another role of mitochondria. This was one role where we did the glycolysis that was outside, but we had the pyruvate coming in, and this pyruvate eventually breakdowns to the ATP. This was one role of mitochondria, but another role of mitochondria occurs when we have protein cytochrome C released from the inner membrane of the electron transport chain. Let's see what does this cytochrome C does. In the cytoplasm, this protein activates set of proteases that degrade all cellular components in a regulated process called apoptosis, which results in the rapid cell death. So cytochrome C that is produced by mitochondria is responsible for apoptosis. Apoptosis is programmed cell death. We will read about apoptosis in detail in just next chapter. But right now we need to know that the cytochrome C release from the mitochondria to the cytoplasm is one important role of mitochondria that results in apoptosis. New mitochondria originates by growth and division of pre-existing mitochondria. We need to remember this, that mitochondria are produced by fission, that is just growth and division from the pre-existing mitochondria. During cell mitosis, each daughter cells receive approximately half the mitochondria in the parent cell. So during cell mitosis, each daughter cell receives approximately half of the mitochondria, of course. Unlike most organelle, mitochondria are partly autonomous of the nuclear genes and activity. So what does this statement means? Means that mitochondria has its own genetic material. This is very important. Let's highlight this. Unlike most organelles, not all of them have their own genetic material. Ribosome does. The mitochondrial matrix contains a smaller circular chromosome of DNA, ribosomes, mRNA, and tRNA, all with similarities to the corresponding bacterial component. Protein synthesis occurs in mitochondria, but because of the reduced amount of mitochondrial DNA, only a small subset of mitochondria. We need to just remember that protein synthesis does occur in mitochondria. This can just be a question. But because of reduced amount of mitochondrial DNA, only a small subset of mitochondrial protein is produced locally. Most are encoded by nuclear DNA and synthesized on free polyribosome of the cytoplasm, the cytosol. These proteins have short-term amino acid sequences that serve as signals for their uptake across the mitochondrial membrane. The observation that mitochondria have certain bacterial characteristics led with the later work to the understanding that mitochondria evolved from the ancestral aerobic prokaryotes that lived symbiotically with an ancestral eukaryotic host cell. So we can just, through all this knowledge that we read so far, make an assumption, or some might have proven it, that the ancestral origin of mitochondria comes from prokaryotes, and now we see a completely developed mitochondria in the eukaryotes. So this was mitochondria, guys. That's it. Let me check if there's anything in the chat. All right, we are already 35 minutes in. Let's get done with the peroxisomes. Very important, and probably the last, I guess, organelle that we need to study. Yes, I was right. All right, peroxisomes. Peroxisomes are spherical organelles enclosed by a single membrane. Remember, single membrane, and named for their enzyme producing and degrading hydrogen peroxide H2O2. So peroxisome is responsible for producing peroxide, hydrogen peroxide. That's why the name is called peroxisomes. Oxidases located here oxidizes substrate by removing hydrogen atom that are transferred to the molecular oxygen producing H2O2. All right. So oxidases, this is the chemical reaction that goes through the oxidases that are located here oxidize substrates by removing hydrogen atom that are transferred by removing hydrogen atom and that are transferred to the water molecule producing H2O2. Peroxides such as catalase immediately break down H2O2, which is potentially damaging the cell. These enzymes also inactivate various potential toxic molecules, including some prescription drugs, particularly in the large and abundant peroxisomes of liver and kidney cells. We need to remember this function as well. So, I don't think we require to remember this chemical reaction here, but it's a basic chemical reaction of production of peroxisomes, hydrogen peroxides. Other diverse enzyme in peroxisomes complete contain complement other diverse enzyme in peroxisome complement certain functions of the smooth endoplasmic reticulum. So just these two names are important, that the enzymes oxidases and catalases, as the name suggests, oxidases, ASE means we know that is going to cleave the oxide molecule, and catalase is going to catalase are basically just break down molecules. Other diverse enzymes in peroxisomes complement contain functions of smooth endoplasmic reticulum and mitochondria in the metabolism of lipid and other molecules. Okay, so this is also important that peroxisomes contain, they complement the function of SER and mitochondria. Functions of SER was to produce steroid hormones. Some protein and steroid hormones are produced, some lipids and steroid hormones are produced by SER. Also some hormones from the P450 cytochrome P450 family are also produced. And degradation, detoxification is also a function of smooth endoplasmic reticulum. And function of mitochondria, as we just read, production of ATP, oxidative phosphorylation in the production of in the form of aerobic respiration and oxidative phosphorylation. Also one more important function of the mitochondria is it secretes P cytochrome that eventually leads to apoptosis. We'll read apoptosis. Thus, the beta oxidation of the long chain fatty acids is preferential accomplished by the peroxisomal enzymes that differ from their mitochondrial compartments. Yeah, we need to remember that the beta oxidation of this long chain fatty acids. So for the lipid, you know, metabolism of lipid is also a function of peroxisome. Certain reactions leading to the formation of bile acid and cholesterol also occur in the peroxisome. This is also one more function, the function of the bile acid, the formation of the bile acid and cholesterol happens in the peroxisome. The bile acid and cholesterol are form of lipids only. So peroxisomes form in two ways. budding of precursor vesicle from the ER, or growth and division of pre-existing peroxisomes. So how are peroxisomes produced? Through just from the vesicles from endoplasmic reticulum, or from the pre-existing peroxisomes. Peroxisomal proteins are synthesized on free polyribosomes and have targeting sequences of amino acid at either terminus, recognition by the receptor located in the peroxisomal membrane, for import into the organelle. So peroxisomal proteins, now as we said that peroxisomes have do produce proteins because they help in the the metabolism of lipid, and on the free polyribosome have targeting sequence of amino acids at either terminus recognized by receptors located in the peroxisomal membrane for import into the organelles. And this was it about the peroxisome. And next up, we are going to start with the cytoskeleton. Let's go in the next lecture to study about the cytoskeleton. And after the cytoskeleton, we are almost, not almost, but we are completely done with this chapter. And we're going to then go into the chapter 3, studying nucleus. So, thank you, everyone.